Good morning, students. Welcome to my online version of lecture. This is the part of the molecular biology of the cell module and mainly aimed for postgraduate students or master students in biomedical science program of Faculty of Medicine Ponorogo University. But if you are an undergraduate student or even doctoral student, feel free to use this lecture on your study. In this lecture, I would like to talk about cell membrane and cellular transport. The main textbook that I use is Molecular Biology of the Cells by Bruce Albert, 6th edition. Okay, as you have already learned during your high school and undergraduate, you would know that all cells have a cell membrane made of proteins and lipids. Some cells have cell membrane, for example, mammalian cells. Some other cells have cell walls, for example, plants, fungi, and bacteria cell. So the difference between the mammalian cells and plants or fungi or bacteria cells are that the mammalian cells, the mammalian cell membrane consists of two layers made of lipid, or what we call as lipid bilayer. The plant cell, in plant cell, they have a cell wall made of cellulose. In our diet, the cellulose is what we call as fiber. While bacteria and fungi also have cell walls, but they do not contain cellulose. Cell wall functions as gate, which is separate the cells from surrounding environment. It is porous and it allows most everything to cross, like a barrier that's able to regulate what's getting into and out of the cell. So in this slide, you can see the cell walls in plant cells and bacterial cells. You can see the different layers from plant cell and bacterial cells. So the function of cell membrane is to separate the components of the cell from its environment that surrounds the cell. It acts like a protection for the cells so that the cell can minimize the effect of its surrounding. For example, from different viscosity and etc. The cell membrane has a characteristic that is selectively permeable. So it regulates the flow of materials from getting in and out of the cell. Some materials can easily go through the cells, and some would need energy or inclusion to the cells through specific protein or channel, and so on. From the homeostasis point of view, cell membrane helps cell maintain the homeostasis, so the cell can have a stable internal balance. We can see the structure of the cell membrane through an electron microscope. So, membrane structure is visible using an electron microscope. And if we would like to see the two layers of membrane, we can use the transmission electron microscope or TEM. If we would like to see the structure of membrane proteins, more details on the cell membranes, we can use the freeze fracturing techniques, which is separate the layers and reveal the membrane protein clearly. Next is about the components of cell membrane. Here is the picture of cell membrane with its components. In cell membrane, we can have phospholipid bilayers and membrane proteins. So phospholipid bilayers means that the layer of phospholipid, which is each of them has phosphate and lipid component, with the phosphate head facing the external and internal environment. The membrane proteins, they consist of several different types. So many of them differ by its location in the cell membrane. Those are, for example, the integral protein or globular protein, which is located across the membrane. So it's facing both in outer and internal environment. There is also surface protein, which is located in the surface, either outer or inner surface. There is also peripheral protein, which is located in peripheral side of the membrane, and integral protein, which its head is both inside and outside of the cell. Here is the structure of the phospholipid bilayers. This is the phosphate head, which is polar and hydrophilic. So it consists of three parts: choline, the green one, and then phosphate, the orange, and glycerol. This hydrophilic part is facing the outer environment, which is the intercellular space or extracellular fluid. And the second layer, the phosphate head facing the inner environment, which is the cell cytoplasm. And then this is the liquid tail. The liquid tail is nonpolar and hydrophobic. So it can function as the barrier for hydrophilic substance, which is able to separate the inner and outer environment. Phospholipid bilayers are fluid, so it means that it can give flexibility for the cells to change its shape and adjust to the environment. So here is the lipid bilayers. One head facing extracellular fluid and another head facing the intracellular fluid or cell cytoplasm. The phospholipid bilayers have hydrogen bond of water, which hold the two layers together, and individual phospholipid and unanchored protein that can move laterally to the membrane. So here's the phospholipid bilayers. The fluidity of phospholipid bilayers is affected by some factors. One, the saturated fatty acid, which make the membrane less fluid than the unsaturated fatty acid. So some cells, they have different composition of the saturated fatty acid or unsaturated fatty acid. The cells which is more on the saturated fatty acid is usually less fluid. The membrane is less fluid compared to those cells which has more unsaturated fatty acid. Other factor is cholesterols. The presence of cholesterols can make the membrane more rigid. And third factor is the temperature of the environment. So warm temperature can make the membrane more fluid than cold temperature. The phospholipid, it has the ability to move laterally, and on a rare occasion, they are able to make a 180-degree turn. So I'd like to talk more details on each component. So now, as we can see, I've already explained the phospholipid part of the membrane, cell membrane. Now I'd like to talk about the membrane proteins. So there are two types in general for the membrane proteins, which is classified by how they are associated with the membrane. As I already explained, the two general classification is the peripheral membrane proteins, which is located on the surface, and the integral membrane proteins, which is inserted to the membrane. So here you can see the peripheral protein, and then the integrated protein, or the globular protein, and also the integral protein, which is alpha helix protein, the surface protein, and peripheral proteins in the sides of the cell membrane. So the fluid, this is the fluid mosaic model of membrane protein. The fluid mosaic model, it represents the consensus view of the cell membrane structure, which is based on the following: that first, the membrane is fluid as the phospholipid have lateral movement. So you can think it as gasoline floating on water or ping pong ball floating on water. The membrane is a mosaic protein which is, can be inserted at various locations of the membrane. So we can insert various protein in here. You can see the phospholipid bilayers with the proteins in between the phospholipid bilayers. The extrinsic proteins are those which is located only on the surface of membrane. Intrinsic proteins are embedded within the membrane. So next is the function of membrane proteins. There are some function of membrane protein. One is the transporters, two is the enzymes. So oxidoreductase, transferase, or hydrolase. The third one, the membrane protein can function as cell surface receptor. Other is as the cell-to-cell addition protein. For example, the membrane protein lectins, integrins, indecans, and caderins. And six is to attach the cells to the cytoskeleton. Now, I'd like to explain first transport protein. This transport protein, it helps the substance to go through the cell, or from inside the cell go to outside of the cells. So transport protein, or we call permeases, it transports the molecules across the membranes. And here, for example, the aquaporins. Aquaporins are special protein channels used to move water across the membrane. So the protein are stuck in the membrane like a mosaic. So it can act like a gate, the transport protein. It can gate, but it can help the molecules to go in and outside the cells. So it's specific for the substance. So the protein transport for glucose, for example, or for the channel ion is specific. It translocates, moves the specific substance through the membrane. It's allow only a certain substance, so it's not one for all. So each transport protein has specific substance which can cross. So the types of transport proteins are: one, the channel proteins with its function by having a hydrophilic channel that certain polar molecule or ions used as a tunnel. For example, the aquaporins, that was channel protein. The second one is carrier protein. The carrier protein function by holding on to their passengers. So they hold the substance which they will pass through the cell membrane. And it can also change shape in a way that shuttles them across the membrane. So it's if channel protein can only close or open, the carrier protein, it can hold the substance, change the shape in a way that shuttles the substance and help the substance to cross the membrane. Channel protein would only allow for passive transport down the concentration gradient, while the carrier protein, it can allow for passive or active, so up the concentration gradient transport. So this is the second difference between channel proteins and carrier protein. Channel protein can only be used for passive transport. Carrier protein can allow for passive and active transport. Other function of protein membrane. The other functions. Oh, it's not one, it's the second. So it can act as an enzyme. So some proteins in the membrane may expose their active site to speed up chemical reaction. The other function is to be a receptor site. So it can act as a receptor. For example, the insulin. Insulin cannot go directly into the cell, but it binds to a receptor on the cell membrane. That's also the protein. So it's other function of the protein membrane is acting as the receptor site. The next function is as the cell-to-cell recognition. So we can differentiate between nerve cells or the myocyte cells or the other cells by looking at the recognition, the cell-to-cell recognition, the marker of the cells. Each cell has its specific marker which can be used to differentiate the types of the cells. The next function is as the intercellular joint. So it can act as the addition of the cells to the intracellular. The last function is to attach to the cytoskeleton or intercellular and also the extracellular matrix. Next, this is the schematic figure of what I have explained. This is function of the cells. This is the transporter, as I've mentioned. It also acts as the passive transport, the ion channel transporter, so as passive transport. It allows it to act like a gate which can allow the specific substance to cross. This is the enzyme which can activate certain substance, become another type of this active form of this substance. Then the cell surface receptor. Some molecules doesn't need to go through inside the cells, but it only acts on the receptor of the cells, and then it produces the effect for the cells. The next is act as cell surface identity marker, which we can differentiate each type of the cells by looking at this identity marker. The next is as cell adhesion and as attachment to the cytoskeleton. So I think that's all for my lecture today. Thank you very much for your attention. If you have any question, you can go to my email address as appear in the last slide. Thank you.